Our next speaker is K.P. Kim. He's a medical oncologist and clinical pharmacologist by training and practice. His work includes precision medicine, PH1 clinics, and GI oncology. He is very involved with the OHDSI Oncology and Genomics work group and leads the effort to add genomic data to the CDM.
Thank you, Melanie, for the kind introduction, and really thank you for the committee for inviting me to give a talk and share my experience on how we wanted to translate really precision medicine into something that would care for the patients. So if you look at precision medicine, actually, we think it's quite similar to the OMAP CDM in that it contains various levels, population, patient level. And not only that, but as I will show, other specimen levels and those kinds of levels all together, and how you're going to integrate all these data to really have an impact on the care of patients. Actually, as precision medicine comes into the era, populations are not the one size fits all or the blockbuster era, but subpopulation stratification, different treatment protocols according to the patient's status, and more to the predictive analytics. In 2017, March, actually, the Korean government did a big step in that they decided to nationally reimburse next-gen sequencing for cancer patients. That did have a very big impact, and for us right now, too, we're sequencing around 50 patients a week. So the amount of data was just accumulating too fast. But we really wanted to have this table or box that we could really contain all this information and have a better impact on patients. But we were also understanding some other points. As next-gen sequencing was entering real world, we did see some differences from when we did it as a research-based study. So when it was researched, the sampling or the procedures were quite homogeneous. It was quite operationable, and you didn't have to think about other things. But with it in the real-world data, it was quite heterogeneous. You had to be really flexible on how you're going to obtain the data and really put it into, link it with clinical data, and how you're going to compare with prior Sanger sequencings or immunohistochemistry data. These were the various things we had to think about, what was the source, when was the next-gen sequencing performed, what was the consent status, and various other assays that were on board. How are you going to all integrate that? But at the end of the day, everything was centered around the specimen, and really, as they say, tissue is always the issue. So if you look at the tables, when we put it into database relationships, we would put the specimen, patient, and a procedure that was put in to obtain these specimens. Was it an operation? Was it a biopsy? And those kinds of things would mix together. And through the genomic meta table, we would put them into basically 4 tables. For single nucleotide variants or indels, we would put it in the mutational table. For copy number variations, we would put it in there. And for translocations or fusion genes, we would put in the structural variants. And for expression, for RNA or protein immunohistochemistry data, we would all put them into the expression. And with these 4 tables, we thought that we would be able to address all these various status of genomic data. As you can see, we built a DB, and I guess at the end of the day, the important thing was that specimen was in the center, and everything was centered around that. And I guess these tables, I won't go through every line of it, but you can just see that they were just basically on how we're going to get the data. And as you can see on the last column, we put in optional. So of course, in the really perfect world or in the research world, you could obtain all these data. However, in the clinical world, depending on vendor, depending on each, the Department of Pathology, there was a variation on which information could be put into the table and which could not be obtained. And expression was another very important part, we thought, because really, how are you going to mix this up together with your prior molecular pathology data was very important to see that it was very consistent across the board. This is a diagram of the data flow of our institute, and as you can see, we were very fortunate that we had the OCS EMR imaging system and tissue bank, REDCap, and the registry data, and genomic lab information system all together, mixed up, and then we would send ETL them all to the data warehouse where they would go on to the CDM, and through the CDM we could be able to capture these clinical phenotypes and genomic data. Some extra issues we did have was, so for these data, we wanted to really go beyond just collecting data and really have an implication in everyday practice. So we wanted to link it with the tissue banks and build an API so that we could build further applications that could be used for precision medicine. This is just an internal validation data that shows that the genomic data that was captured by the CDM and our clinical database, that was the GLIS, there was showed identity. And for external validation, we chose two things. So for TCGA, we used it so that we can see that really is the genomic landscape similar. The diagram on the left shows colon cancer and the right shows lung cancer, and easily colon cancer, as expected, was quite similar, while lung cancer had an East Asian flavor to it that differed from TCGA with higher incidence of EGFR. Another thing we did was to see how we were doing operationally, and an institute, Bundang Seoul National, really helped us out on that part, and to see how the data was captured within their sequence and really how it could be, that the tables were quite nicely functional. We went on to build applications to really see how this would get, how we could easily identify it at the population level. At the patient level, you might not be able to see, but really how treatment was put in, which was first-line regimens, what was second-line regimens, and what other immunohistochemistry data were all on board. And then for next-gen sequencing, sometimes you would do a re-biopsy or another next-gen sequencing again, so how that would compare what was concordant, what was discordant, and at the specimen level too. For the tissue bank, for this tissue, is there plasma, or is there buffy coat, or is there normal tissue, or tumor tissue, and what was the quantity of it? You could all just easily confirm. These were some of the points we had to think, consents and other cancer CDM specific issues that we think should be more evaluated by the community. And these are my acknowledgements. Thank you very much.